And now I'd like to introduce another RR program member, my colleague Faye Rogers, and she's going to be talking about targeting amplified gene loci as a pro-apoptotic anti-cancer therapy.
I'll just make sure you're good. Oh, okay, we're good. Thank you, Joanne. So today I'm going to talk to you a little bit about a project that we've been doing in our lab. I'm shorter than Jung, so. Okay, sorry about that. So amplification of genes involved in normal cell growth and survival pathways drive oncogenesis in a broad spectrum of tumors, and which ultimately affects tumor progression and clinical outcome. So as a result of this, it would be the ability to target gene amplification or its protein products gives enormous potential for drug development. So today I want to talk to you a little bit about some technology we've been developing in the lab where we are trying to find novel ways to treat cancers that are characterized by gene amplification. So I have no disclosures. So gene amplification is defined as an increase in copy number of a restricted region of a chromosomal arm. And several oncogenes are amplified, which leads to increased gene expression and gives cancer cells the ability to grow more aggressively or to become drug resistant. So gene amplification happens somatically by various types of mechanisms, one of them being breakage fusion bridge cycles, which actually causes an increase in the number of discrete regions of the chromosomal arms. These amplified regions can either take place on the original parental arm, they can translocate to another chromosome, or they can result in extra chromosomal elements. And these different amplified regions can either be kilobases or megabases long, so which could result in not only having the amplified genes, but as well as passenger genes. So these different amplified copies give a really strong mechanism for why many cancers actually thrive. So as I said to you, oncogene amplification is found in a broad spectrum of cancers, and is considered to play a very major role in not only tumor progression due to its ability to increase cell proliferation or to decrease apoptosis, but also can aid in the transition to metastasis because of increased angiogenesis or to help mitigate migration and invasion. So this increase in DNA copy number actually serves as a major mechanism for driving oncogenesis overall in a broad spectrum. So it turns out that gene amplification is actually pretty frequent in tumor evolution. Analysis of large-scale databases like TCGA have identified over 461 genes that are amplified over 14 different types of cancers. And we can see here a list of cancers where we can actually have gene amplification. And you can see that we can have gene amplification, let me use my thing, in the brain, cancers, breast, lung, gastric, colorectal, ovarian, and melanoma. So as you can see, there are a wide range. So these amplified genes represent a number of different gene families, including epigenetic regulators, cell cycle-associated genes, DNA damage response, and repair genes. So there's a wide array. So because of its importance in tumor progression, a number of drugs have been developed to inhibit the oncogenic activity of amplified genes. However, the majority of these therapies actually have been targeted towards the overexpressed protein product. The first drugs that have actually been directed towards these overexpressed proteins have made enormous impact in cancer therapies. However, there has been some limitation because drug efficacy has been limited a little bit primarily by drug resistance. So one of probably the most beneficial examples of cancer therapies that have been used to target gene amplification has been Herceptin or trastuzumab, which is a humanized monoclonal antibody that targets the extracellular domain of the HER2 receptor. Herceptin disrupts HER2 downstream signaling and has been used in the treatment for HER2-positive breast cancers and has made an enormous impact. And some people will tell you that because of Herceptin, we've had almost a cure for HER2-positive breast cancers. But unfortunately, primary and acquired resistance does occur in some of these patients. So there still is a need to find new therapies that can be used for the treatment of these particular cancers. So my lab primarily is interested in doing DNA repair and DNA damage response. So our thought was, would it be possible for us to find a new way to treat cancers that are characterized by gene amplification by manipulating this important pathways that are important for maintaining genomic integrity? So DNA damage response is made up of several different pathways. If a cell is introduced with DNA damage, either from endogenous or exogenous agents, the cell can choose to repair that damage by activating checkpoint activations, and that repaired DNA damage can lead to normal cells. However, if the damage is too extensive, the cells can choose to activate another pathway, which leads to apoptosis and preservation of genomic instability, preserving of genomic integrity, because if you have excessive damage that is not repaired, you can have genomic instability, which can lead to enhanced carcinogenesis. So this balance between DNA repair and apoptosis is very important for preserving genomic integrity. So the thought for our lab, since we've been working extensively in this pathway, is it possible for us to manipulate this pathway in order to find new technologies that can be used to treat cancers that are characterized by gene amplification? So the favorite pathway in my lab that we work on is the nucleotide excision repair pathway. It's important for the recognition and repair of a wide array of helix distorting lesions. And the first important step of nucleotide excision repair is damage recognition. This happens by two separate proteins, depending on what part of the genome this damage occurs. If it happens in an actively transcribed region, it is recognized by the protein CSB, which then activates the transcription-coupled repair arm of this pathway. If it's within the, just anywhere within the genome, XPA and XPC play a very important role in activation of global genome repair. This then leads to the recruitment of repair complexes that unwind by different helixes. We have incision, gap filling, and then repair. So I think that most people probably would agree that nucleotide excision repair is probably one of the most versatile DNA repair pathways. So our thought was, could we possibly manipulate nucleotide excision repair to create a technology where we can force the cells to either repair or activate apoptosis in cancers that are characterized by gene amplification? So the question is, can we then develop a platform that directly converts amplified gene loci into DNA damage that can then activate the cell to trigger its own death and activate cell apoptosis? So our approach was to use triplex-forming oligonucleotides. So triplex DNA can be formed when oligonucleotides bind in the major groove within a sequence-specific manner. So these structures can occur at a polypurine, polypyrimidine stretches, and they can either bind parallel or anti-parallel, either in the purine or pyrimidine motif. And so sequences capable of being targeted by triplex-forming oligonucleotides happen as frequently in the genome as 1 in every 50,000 base pairs. And so if you look by bioinformatics, it actually turns out if you map how many polypurine-rich sequences that can be found, there are over 519,000 unique triplex-forming sites within the human genome. And it turns out that each gene has at least one unique site to that particular gene that is capable of being targeted by a triplex-forming oligonucleotide. So as a result, you would expect that every amplified driver cancer gene should have some unique triplex targeting site that one could design a triplex-forming oligonucleotide to bind specifically to that gene of interest. So the first thing that one would want to make sure that they can ensure is that, are these triplex structures actually repaired? Because in order to manipulate that pathway DNA damage response, we have to first ensure that we can have repair. So to do this, we used an in vitro repair assay where we incubated 2 different plasmids, one with and without the triplex target site. We designed a triplex to bind to the target region, and then in a ATP regenerating system with radio labeled nucleotides, we then look to see for incorporation of that radioactivity to see as a significant, as a representative of repair synthesis. And so as you can see with our triplex that we designed to target this reporter gene, that we can see an increase in the amount of radioactivity incorporated in its presence. We then went on to do other activities to then go on to prove that this repair synthesis is dependent on nucleotide excision repair. So we knew that triplex formation was recognized by the cell as DNA damage, and this was dependent on nucleotide excision repair. So the next thing we wanted to go on to prove was, is it possible for these structures to actually cause a cell to induce enough DNA damage to activate apoptosis? So to do this, we generated a cell line that had multiple copies of our triplex binding site. And we found that if we incubated with our triplex, we could see that we have a loss of cells growth. We also used annexin 5 as a measurement of activation of apoptosis, and looked by Western blot, by cleave-parked and activated caspase-3 as another marker for apoptosis. And so this was then confirmed in an in vivo mouse model to show that we could also see these apoptosis being activated in mice as stained by caspase-3. So this kind of confirmed our belief that we could use triplex induce DNA damage as a means to not only induce DNA repair, but if enough of the triplex structures are around, we could also activate apoptosis. So we wanted to focus our initial work on cancers driven by HER2 amplification. This being because it's a very good model system. We already know that drugs targeted to HER2 amplification work extremely well. As I said to you before, Herceptin and the drugs that follow work very well in the clinic. And so just to give you a little bit of background, HER2 is a receptor that's normally involved in cell transduction pathways and important for cell growth and differentiation. And its overexpression provides tumors with the ability to grow pretty aggressively. So about 25 to 30% of breast cancers are HER2 positive. And this overexpression is due to gene amplification. And so one would have also found that this overexpression correlates to enhanced tumor aggressiveness, greater likelihood of having recurrence, and a decreased inpatient survival. So we decided that we would use this HER2 positive breast cancer as a model to determine whether or not our strategy was effective. So just to kind of describe our drug design scheme, so what we imagine is that in a normal breast cell, that you would only have 2 copies of the HER2 gene, while in the amplified cell, you would have multiple copies of the HER2 gene. If we design HER2-targeted TFOs, they could bind in the normal cell to only 2 copies of this HER2 gene to form a triplex. This would be recognized as damage by the nucleotide excision repair pathway. The nucleotide excision repair pathway would repair the damage, leaving you with a normal cell, and the cell will survive. However, in the case of the amplified breast cancer that has multiple copies of the HER2 gene, you would have excessive DNA damage, which would lead the cell to activate apoptosis. So this would give you a way to specifically target the amplified cancers, while the normal tissue should have minimal toxicity. So to test our hypothesis, we designed 2 TFOs to target the HER2 gene on chromosome 17, one within the promoter region and the other within the coding region at position 205 relative to the start codon. And then in order to determine that we were actually having a binding event, we used non-denatured metaphase spreads. And you can see here, green would be a marker for chromosome 17, which is the location for HER2. Red would be our labeled oligonucleotide that's tagged with TAMRA. And so you can see for the MCF7 cells that only have 2 copies of the chromosome 17, we can only see 2 binding events, while in the BT474 cells, which have multiple copies of the chromosome 17 site, you can see we have multiple binding events with our targeted TFO. It's kind of showing that we do have sequence-specific binding of our oligonucleotide to our gene of interest. So then first thing we needed to determine once we know that we have reagents that bind to our site of interest was to then test to see whether or not we can induce DNA damage. So we used a range of breast cancer cell lines that had different variant copies of the HER2 gene, leading from cell lines that had no copies to ones that had up to 52 copies of the HER2 gene. And this just kind of Western blot to show that these the HER2 protein is amplified in these particular cancers. Cell lines. And so we use a neutral comment assay to look to see whether or not we were having an increase in DNA damage in a variety of different cell lines. And you can see that with our HER2 targeted TFO, we have increasing amounts of tail moments signifying DNA double-strand breaks. And if we look at this in comparing the amount of DNA damage compared to the copy number, we can see that with an increasing amount of copy numbers, we have an increase in the amount of DNA damage, suggesting that our TFOs are binding specifically to a site of interest, and this damage is dependent on copy number. So just to confirm that we were also having double-strand breaks, we did gamma H2X positive cells staining and quantified this via FACS, which also confirmed an increase in the amount of DNA double-strand breaks in the presence of our TFO, also by looking at immunofluorescence and quantification of both gamma H2X and 53BP1, which also confirmed that we were having an increase in double-strand breaks. So now we can say that we were able to create TFOs that were able to have an increase in the amount of DNA double-strand breaks that's copy number dependent. But the question was, can we then activate apoptosis because of this damage? So we also went ahead to measure the amount of apoptosis, and we found in several breast cancer cell lines, as measured by Cleve-PARP and by looking at Nexin 5 positive staining, that we do have an increase in the amount of apoptotic cells following treatment with our HER2 targeted TFOs. This once quantified, we can then confirm that this level of induced apoptosis was dependent on copy number. So suggesting that we were able to create DNA targeting molecules that were sequence specific and that could induce apoptosis that was dependent on the amount of HER2 copy sites. So in order to see whether or not this had any therapeutic efficacy, we used a human xenograft model for BT474 cells. This particular model had been used to originally test the efficacy of Herceptin, which I said to you previously is the currently used targeted therapy for HER2-positive breast cancer patients. And we can see after 20 mg per kilograms of our TFO, we see a decrease in the tumor growth. Also, we use Herceptin as a control. And using Kaplan-Meier curve, we can see that the drug efficacy between our HER2-targeted TFO and Herceptin was similar, which really was very promising for us to see that we were able to create molecules that used a different mechanism of action to give the same level of drug efficacy as we see with a currently used drug. Using immunohistochemistry, we can see that we can see by caspase-3, we can have apoptosis in our tumor. See also by looking at HER2 to see that this is a HER2 positive. Also, if we take a look, we can see that this area of the tumor is actually infiltrated by neutrophils and macrophages. So one of the things we were also interested to look at was to use another mouse model for ovarian HER2-positive cancers, because it turns out the HER2-positive ovarian cancers do not respond very well to the targeted therapies that have worked so well for the HER2-positive breast cancers. And so we used two ovarian cancer cell lines that have HER2 over-amplification, and this is just FISH showing that there is amplification. And then look to see whether or not we were able to induce DNA damage in these cells. and then looked to see whether or not we were increasing the amount of apoptosis in these cell lines, which we saw the same thing that we did see with our breast cancer cell lines. And then went on to do a mouse model for SKOV3, ovarian cancer cell lines. It turns out that because these HER2-targeted therapies don't work very well for ovarian, the method of treatment currently is to use drugs like cisplatin, which always have a problem with drug resistance. So this particular ovarian cancer cell line is actually resistant to cisplatin, and you can see cisplatin resistance, but we did still have, our TFO was able to reduce growth of our tumor xenografts, which was really encouraging for us. So kind of what was really important is that our whole technology is that we are using a different means of inducing cell death into these gene amplified tumors, while we want to prove that this is not a result of us manipulating gene expression or HER2 function. So we did RT-PCR to look at gene expression and then also look at phosphorylation status of both HER2, and we see that our active TFOs were not affecting HER2 protein expression in any way, suggesting that our method of drug activity was independent of HER2 function. We went on to confirm that we also were not affecting any of the HER2 families proteins, either HER2, 3, 4, or EGFR in their phosphorylation events, also further confirming that whatever the TFO was doing was independent of HER2 activity. So as I said to you before, our thought is that we could use DNA damage response to actually activate tumor-specific apoptosis. So we went on to prove that this was indeed the mechanism of action by looking at some common DNA damage response protein, by looking at the phosphorylation status of CHEK1, CHEK2, and we've also looked at phosphorylation of ATM, and we found that after following treatment with our HER2-targeted TFO, that we do have an increase in phosphorylation of event of DNA damage response proteins. So to look a little bit more in depth, as I said to you before, there is this delicate balance between DNA repair and apoptosis, and it's been shown, and something that my lab is working on, is that some of the repair proteins that are involved actually play a major role in activating apoptosis. And we're interested in a nucleotide excision repair. And previous studies had shown that XPD was important for activating apoptosis following nucleotide excision repair that was dependent on p53. So we wanted to see whether or not this could be the case for our HER2-targeted TFOs. So we knocked down XPD and then looked for activation of apoptosis by measuring cleaved PARP, and found that if we knocked down XPD, we no longer had activation of apoptosis. And then wanted to confirm, was this loss of apoptosis also dependent on p53? And so we knocked down p53, but what we found was that we still had apoptosis. So whatever mechanism was being activated was dependent on XPD, but independent on p53, which in terms of cancer is actually a good thing, since most cancers are p53 either null or mutant. So that was exciting news. So we've taken steps to kind of figure out what could potentially be happening. So it's been previously reported in the literature that gamma H2X actually has two phosphorylation sites, one at serine 139, and in addition to that, one at tyrosine 142. And what studies have shown is that phosphorylation events at both the serine 139 and tyrosine 142 need to take place in order for the activation of apoptosis. So we looked to discover what the phosphorylation events were after our TFO treatment, and we found that we did have dual phosphorylation at both serine 139 and tyrosine 142, suggesting that our TFOs were activating this particular mechanism. And then in looking, we did some proximity ligation assays, and we found that XPD was interacting with the tyrosine 142 phosphorylation site on gamma H2X. And so what we found that is if we knocked down XPD, we didn't have apoptosis, but we also didn't have phosphorylation of that tyrosine 142 site, which would suggest that XPD was required for that phosphorylation event. And so this is probably what P53 independent mechanism we were actually activating. So we went on to further confirm that if we knocked down P53, we found we still have apoptosis, we still have serine 139 phosphorylation, meaning that we still have DNA damage, and we still have phosphorylation of tyrosine 142, which would suggest that we have been able to utilize a novel mechanism of action to induce DNA damage and a apoptotic event that's specific for cancers that are characterized by gene amplification. So a working mechanism of action is that we can design triplexes specific to our gene of interest. This can lead through replication stress. We know this from other work that we've done to double-strand breaks. These double-strand breaks activate DNA damage response, leading to both phosphorylation of ATM, check 1, check 2, and most importantly, that we have this dual phosphorylation both at serine 139 and tyrosine 142 that activate a P53 independent pathway that is dependent on the presence of XPD. So just to kind of quickly summarize what I think hopefully you gathered today is that we were able to design TFOs that target polypurine sites that are specific for the HER2 gene, and that these TFOs were able to activate gene-specific apoptosis, and specifically in HER2 overexpressing cancers, and that this apoptosis activation is dependent on copy number. And so that also our mechanism of action is independent of HER2 protein function, but dependent on that DNA damage response. And so just to kind of quickly tell you what we're working on right now, so we know that drug delivery will be very important for improving drug efficacy. So we've started a collaboration with the Salzman lab. And just in a quick experiments, we have found that if we load our TFOs into nanoparticles, that we can have, after one dose, a pretty dramatic decrease in tumor size. And in addition, we have been exploring work showing that DNA repair deficiencies can also increase the activity of apoptosis induced by TFOs. In using ovarian cancer cells that are BRCA proficient and deficient, we find that not only can we increase the DNA damage, but also have an increase in the amount of apoptosis. So we think that as a means to kind of improve the drug efficacy, we can not only look at improving drug delivery, but if we can manipulate DNA repair in some ways, that we can also be able to boost the efficacy. Just to thank the people in my lab, a lot of this work has been done by Mitu Kashik and Cynthia Chan, and all the funding and our collaborators.